This recorded lecture is dedicated to part 1 for module number 5 on fungi as human pathogens. I have decided to divide this session into two. The first part will be recorded. We'll talk about the general characteristics, the general biology of fungi. We'll cover the pathogenic and the non-pathogenic groups. And then during our on-site lecture tomorrow and on Tuesday for all the block sections, I'll talk about the medically significant fungi and their pathogenesis. Okay? So, this is the outline for this module. So, we'll cover, for this recorded session, we'll cover the general characteristics until the major importance or significance of the fungal group in different industries. Okay. Typical examples of fungal organisms include yeasts, molds, and macroscopic mushrooms. Yeasts are unicellular fungi, which are known to have significance in the food industry. Medically, there are known yeasts that cause diseases to human hosts. Molds, on the other hand, are filamentous fungi. When they are grown on fungal specific media, we can appreciate that they can be seen as leathery colonies. And microscopically, when you observe them, you get to appreciate their filamentous nature. Mushrooms, on the other hand, are mostly are macroscopic. Some are edible, some are known to be toxic to human organisms, no? Or to human hosts. We'll cover mostly yeast and molds for the medically significant representatives during our on-site lecture. Now, a review on the biology of fungi, comparing it against bacteria as a major group of human pathogens. In terms of the cell type, fungi are eukaryotic, unlike bacteria, which are prokaryotic. Fungal cells have compartments within them, organelles such as the mitochondria possessing its own DNA, endoplasmic reticulum facilitating secretory processes and protein translation processes. Unlike in bacteria, where those structures are absent, no? In terms of the cytoplasmic membrane, fungi possess sterols, allowing for um, modified fluidity, unlike in bacteria, where the cell membrane, the cytoplasmic membrane, do not have sterols. The cell wall is also another distinguishing feature between these two groups. Cell wall of fungi are primarily made up of chitin. Some species may possess other types of compounds such as glucans and mannans, while for bacteria, mostly what we know about gram positives and gram negatives is the difference in their peptidoglycan layer and their murine layer. Additional macromolecules can also be found in the bacterial cell wall, such as teichoic acids for gram-positive bacteria, glycolipids for gram-negative bacteria, the lipopolysaccharide, you remember that very well at this point, and some proteins, important for cell signaling. In terms of metabolism, generally both groups are heterotrophic by nature. Some bacteria are autotrophic, some photosynthetic representatives, while for fungi, they're heterotrophic. No species are known to harness light energy for the production of carbohydrate substrates, unlike the autotrophic bacteria. And a key feature or a key characteristic is a lot of fungi are known as saprophytes. They adapt a lifestyle where they consume material from the substrate that they grow onto. In terms of oxygen requirement, most fungi are aerobic, unlike bacteria. There are species that are either obligate anaerobics or species that are obligate aerobics, requiring oxygen all the time. And some are actually facultative, switching oxygen requirement routes. No? Some can grow facultative anaerobic bacteria can grow best in the presence of oxygen, but they can also switch to a lifestyle that is anoxic. In terms of size, generally fungal cells are bigger, 3 to 10 microns in size, unlike bacterial cells, which are generally small, 1 to 5 microns. And in terms of dimorphism, some fungal species are dimorphic, some species of them, while bacteria are no representatives are known as dimorphic. Okay, so we classify fungi into three major groups: the true fungi, the Eumycota, the Straminifila, and the slime molds. So we'll cover mostly the Eumycota, that has further subgroupings: the chytrids, the zygomycetes, the glomeromycetes, the ascomycetes, the basidiomycetes, and what was previously known as Deuteromycota, the mytosporic fungi, also known as fungi imperfecti. So let's take a look at these classifications. So chytrids are under Chytridiomycota, are the basal group, the basal classification of Eumycota. Zygomycetes mostly are the bread molds. Okay, so they colonize carbohydrate-rich substrates. No? Then there is also arbuscular mycorrhizal fungi under Glomeromycota, associated with environmental sites or environmental sources, like soil mostly. Then there is sac fungi, the Ascomycota. The prefix asco or ascus means sac or sac-like in appearance. And we have the Basidiomycota, which are the club fungi. We won't really cover the imperfecti group, the fungi imperfecti group in this lecture. Okay, so a few more things about the biology of fungi. They are cosmopolitan in distribution. They appear everywhere. No? About 200 of the thousands of species identified are are pathogenic to humans, a very small portion. And a lot are actually or more than, less than a dozen of these known fungal pathogens the cause most of the documented human diseases or human infections. In medical mycology, fungi classified according to practical aspects are known as are into further classifications. No? Yung ating dermatophytes, the ones that cause skin diseases, the yeasts, molds, and dimorphic fungi. Okay. I already mentioned about their heterotrophic lifestyle and their saprophytic lifestyle. No? In terms of their life cycle or in terms of their existence, some fungi can exist in both saprophytic nature and in biotrophic nature. Okay? And we generally classify them according to morphology. So there's a lot, in terms of classification, we really have to grow them in the lab to be able to justify their identity or the taxonomic assignment that we give to them. No? In terms of shape, there are unicellular fungi, the yeasts, the filamentous ones, the molds, and for the basal groups, we have those that are possessing characters in between plants and animals, those that have flagella, are motile, capable of swimming, and some species that are able to insist in order to form sporangia. Okay, so now looking at the general characteristics, let's examine let's examine the group further. So all are known as eukaryotic, okay, possessing DNA bound by a nuclear membrane. So there's a specific nucleus carrying the genetic material of the organism. There are also structures such as the mitochondria, ribosomes, facilitating processes such as ATP production and protein translation. And then there's a cell wall, primarily made up of chitin. In terms of their gross morphology, gross and microscopic morphology, so molds I mentioned are already are mostly filamentous, forming a complex structure called the mycelium. So when you grow them in the laboratory, you appreciate the colonies appearing as leathery in gross morphology. And some are unicellular for the case of yeasts. Fungi, mushrooms, on the other hand, mostly are, or I mean, all are macroscopic. The mushroom group, no. Okay, so in terms of the unicellular organization, okay, so let's take a look at molds as a representative group. The protoplasm of a hypa or a cell is surrounded by a cell wall made of chitin and other molecules. So here you can see a hypa, mostly based on the appearance of this picture or this the representative fungal species. No, this is probably a sexual hypa because there is a structure, a spore-bearing structure that is supported by it. Protoplasm here is you can appreciate the protoplasm is surrounded by a cell membrane and a cell wall. Okay. Now, in terms of reproduction, many fungi are capable of sexual and asexual reproduction. So, key difference is in asexual reproduction, there is no recombination of opposite types or opposite mates of the fungal species. Asexual meaning the parent cell is genetically the same with the offspring or progeny cell. And key examples of fungal groups that carry out asexual reproduction are the yeasts. On the other hand, sexual reproduction is well documented in they call this well documented in filamentous fungi in molds. No? So in sexual reproduction, there is meeting or there's meeting and union of opposite mating types. No? And that will result to fertilization. During and after fertilization, the spore will undergo meiosis to make sure genetic diversity is increased, and these spores, as a product of sexual reproduction, can carry out or can produce a new individual mold once grown or once it finds a suitable substrate. Okay, right. Then for the nutrient acquisition, fungi are chemoheterotropic, organotrophic. All are void of chlorophyll, so they're not able to carry out photosynthesis. Okay? They're not able to harness light energy for them to produce their own nutrients. Mainly saprophytic, they get energy from dead organic matter. Okay? They possess characteristic range of storage compounds. So they can produce inclusion bodies that contain reserve molecules. They can be free living or they may be capable of forming relationships or symbiotic or not with other non-fungi organisms. Okay, so here let's examine further the morphology of fungi since, as I've already mentioned, Taxonomy, classification of fungal species is highly dependent on known morphological documentation. No? So the body or the soma of the fungi is called the thallus. For the case of molds, filaments exhibiting growth, no, tubular growth, are known as hypa. And these hypa, when clumped together, forms mycelia. Okay. Right, so based on septation, the fungal hyphae can be septated or not. So there is division or a septa within the hypa that divides it into short portions or segments. So that's septated hypa, as you can see in letter A. And in letter B, you see that the hypa is sinusitic or non-septated. So there are no septum or walls that divide the entire hypa, into small segments. Based on function, the hypa can be of different types. Some are vegetative that function for carrying out nutrient acquisition. They penetrate these types of hypa, penetrate the media or the substrate to absorb food material. There is aerial hypa, which are directed above the surface of the media or the substrate where the mold or the fungi is growing onto. There is reproductive hypa, I think I mentioned this a while ago, which are aerial hypa that carry different spores. They are supporting spore forming structures. And combined together, all of these hypa types are comprising the mycelium. Okay, so here you can see Aspergillus niger. There is vegetative hypa, primarily subsurface in orientation. There's also aerial hypa growing onto the surface or onto the open surface, supporting structures such as structures such as the spore-bearing structure. So here, when grown on fungal-specific agar, you can see aerial hypa growing above surface of the culture medium. Okay. In other types of associations, no, fungi species can also produce different kinds of structures. Hostoria is one of them. So a hostoria or a hostorium is an outgrowth of the fungal hypa onto the cells of the organism that they are forming intimate relationship with. So Hostoria is typical in plant fungi associations. So here you can see the Hostoria growing onto the plant cell. And some of them can be very parasitic, gaining nutrients produced by the associated plant species or the plant cell. Okay, here is a more vivid example. The hypal tip penetrating the host tissue and draws nutrients from it. Okay, so this is the hypa, and then there are hostorium growing onto the plant cell. These are the hostorium projections or hostoria projections. Okay. In fungi, in molds, no, that grow on substrates like bread, for example, the known bread mold, Rhizopus sp. So they form root-like structures called rhizoids. So these are vegetative hypa growing below or within the substrate. There's also stolon, a hypa that connects two rhizoids. So here you can see stolon connecting rhizoids or separate modules of the fungal mold. No? Okay. In cases of an interesting other type of structure produced by fungi is a constricting ring that is formed by a fungi that can parasitize on eukaryotic multicellular worms like a nematode, no? So this constricting ring, as you can see in this scanning electron micrograph, okay, it constricts the nematode organism. So here you can see the constricting ring, okay? So this one, a specific species. So we have Arthrobotrys oligospora, which can capture a nematode when it merely touches the outside of its trap. So the fungi, this fungi is known to parasitize on um, nematode organisms. So in terms of nutrition, so I've mentioned this already. Some species of fungi can also be phagocytic. Primarily, the reason behind or not reason, but um, the logic is as chemoheterotrophs, they source their carbon, nitrogen requirements from the the organism that they grow onto or the substrate that they grow onto. Okay. In terms of temperature requirement, so fungal species grow in a spectrum. Some can grow in very low temperatures, others in temperatures up to 40-39 degrees Celsius. So mostly are grown in mesophilic conditions, so 37, 38 to 39. In terms of pH requirement, okay, most fungi grow in neutral pH. Others actually prefer a more basic pH, and others can also prefer acidic conditions, especially for the case of yeasts. No? As obligate aerobes, so they require oxygen as eukaryotes, no? And others can also be requiring high concentration of salts in their medium, from their ambient. Okay. Now, many fungi are capable of also forming symbiotic relationships with plants. So lichens are associations between fungi and plant-like organisms, algae and cyanobacteria. Algae or cyanobacteria. So the fungi serves as the mycobiont, while the cyanobacteria is the photobiont. So the photobiont is responsible for photosynthesis, for the formation of nutrients from raw sources, while mycobionts, on the other hand, the fungi group, shelters within the algae and they collect minerals and water. You can collect mostly the form products by the cyanobacteria. So some examples of lichens, so those that grow onto solid surfaces such as rocks. You can see here, these are lichenous growths. Okay, so structurally, so you can see the algal cells, the spherical green colored structures here, form intimate relationship with the fruiting body of the fungi. Okay, so some more examples. We have a crustose lichen growing on a wall, a foliose lichen growing on a tree, on a branch of tree. No, this one is an example of more of like a plant-like morphology for lichens. So this is wolf lichen, Litharia bufina, which grows like which appears like a plant, no, para siyang leaf, no, or shrub in appearance. It has what we call a fruticose growth formation or morphology. This one, on the other hand, is Caloplaca talincola. This lichen grows like a cross and in a pattern radiating outward. So you can see from the center. So the if you look at the outer edges or appreciate that there's an outgrowth pattern, no, forming crustose growth form. Other types of lichens include this one. The species Chrysotrix chlorina grows like a powder that is dusted on the rock surface where it's growing onto. So the appearance or the growth type is leprose in appearance. And some are gelatinous like Collema nigrescens, which do not really have internal structure for its parts, so it's jelly-like in appearance. Okay, so asexual reproduction is important for species propagation in most fungi. And in fact, in most conditions, asexual reproduction is the route or the life cycle that is being followed because it's less energy requiring, it's faster, it's more efficient for the fungi, no? And it results in the production of multiple individuals, sexual reproduction. The thing is, there is no genetic diversity. So you expect that the progeny fungi are genetically the same with the parent fungi. Okay. Okay. So what are the different methods for sexual reproduction of fungal organisms? We have fragmentation. The hyphae break up into component cells that can behave as spores. And these spores, arthrospores as an example, can give rise to an individual, to a new individual. There's also fission, which is simple splitting of a cell into two daughter cells by constriction and formation of a ring, no? A contractile ring. So as an example, we have here yeast undergoing fission. Okay. Budding is also another type of asexual reproduction. Small outgrowths emanate from the parent cell, as you can see here. So this outgrowth here, and this one as well. And the cyan-colored outgrowths here in the from the green-colored yeast cell. This is budding. Okay. Then spores, most common method for reproduction, for asexual reproduction. So the fungal spores, they vary in color, in shape, in size, in their total number as released by a spore-forming structure and their arrangement. Okay. Now, the different types of asexual spores, let's cover them. So we have sporangiospores, which are born within the structure called a sporangium. So some of them are motile with flagella. So those are zoospores. And some are non-motile, the aplanospores, which are not born with flagella. Then we also have blastospores, which are produced via budding for the case of yeast, like for the case of I think some what do you call these fungi? So some um, I think non-sporangium bearing fungal species, blastospores, uh, produced by I think mostly yeast and some fungal molds. No? They have blastospores as a type of an asexual spore. Then for conidium bearing, no, or conidia bearing molds, so they produce conidiospores, which are not really housed within the structure, unlike sporangiospores. So conidiospores are exogenous; they're naked. They can be freely released. A good example of this are the aspergillus. They produce conidiospores. So we have sporangiospores, conidiospores, and blastospores. Now, spores germinate once that they find a suitable substrate or a suitable growth pedestal, no, or growth platform. So the spores allow for the growth of a hypa, and eventually this hypa can, provided that all conditions are optimal for its germination, will give rise to an individual fungal organism. Okay. Now, in terms of sexual reproduction, there are three phases of sexual reproduction in fungal species. There is plasmogamy, karyogamy, and meiosis. Plasmogamy is when there is union of two protoplasts bringing together nuclei that from the opposite mating types within the same cell. Then these mating types that are brought together, they undergo karyogamy, where the nuclei fuse as one. No? Then meiosis is when there is reduction of the number of chromosomes to haploid, because after karyogamy, you expect the reproductive structure to be diploid. No? So it will now carry out meiosis to increase genetic diversity, or to change the genetic makeup of the progeny fungi, the progeny spores. Okay. So here, you can appreciate the life cycle of a fungi following a sexual, following sexual reproduction. So sexual and asexual reproduction are tightly connected. So during sexual reproduction, the end product will be spores that can be within structures such as the sporangium, or can be free within the mycelium. And then these spores can carry out asexual reproduction. No need for meiosis. Okay. This is another example, another visual aid to help us understand the intimate relationship or connectivity between sexual and asexual modes of fungal reproduction. Okay? Fungi are important in different fields, in different no, no, in different applications. So industrially, some fungi are known to be important for making alcohols, for making wine and beer, for fermenting products that are rich in sugar, rich in dairy. No, some can also produce fine chemicals like drugs. Cortisone is an example of material, fine chemical produced by a fungi, and some can also be capable of producing vitamins. And I think the most popular industrial application of fungi is their capacity to be harnessed, or our capacity to harness them for antibiotic production. Okay? So cheese or cheese fermentation. So there are known fungi capable of assisting cheese fermentation. So as an example, we have Penicillium roqueforti, responsible for the flavor profile of Roquefort cheese. Then we also have for Gorgonzola, you can see the fungal molds that are growing onto it, giving it a very different or very unique flavor profile. As food, macroscopic fungi, mushrooms are known as alternative sources of protein. No? Others also, for the case of fermenting liquid products like soy sauce, no? And miso. Okay. Here in some at some other examples of some other examples of fungal-based foods. Not really familiar with them, so some examples include willa in North America, as a food material. In Northern Europe, so there is Iceland moss, and then in Oriental regions like in Korea and Japan, so some fungal source foods are also available. Okay, this one is an example: Korean pan-fried Umbilicaria esculenta, and this one Iceland moss, which is also fungal. No? In terms of fertility of the environment, particularly the soil, no, as saprophytes, fungi actually are important as recyclers of many chemical elements. No, so they take up nutrients from dead organisms, allow them to make use of they make use of these nutrients, and essentially they allow for dissolution of complex macromolecules into simple ones, and these simple ones now become available to other organisms or the matrix itself, like for the case of soil. Okay, so a good example is mycorrhizae, which is an association between plant roots and fungi, allowing for nutrient availability to be not a problem for the case of growing plants with associated mycorrhizae. Okay, so in our Okay, other examples before we talk about the medical significance. So in other applications, some fungi are known as degradatory species capable of degrading macromolecules like resins. No, some are also capable of, I think, being applied for bioremediation of heavy metals. Others are used for production of commercially important dyes, pH indicators, and I already mentioned about antibiotics. No? And then medically, or more of the safety aspect, no? Some fungi are known to possess destructive effects, no, or destructive roles to humans. Some can carry out disintegration of organic matter, mostly are capable of spoiling food material, and of course pathogenesis, no? Diseases in different groups, in plants, animals, and in humans. So we'll start with this slide on my on-site lecture tomorrow and on Tuesday. So we'll talk about fungal pathogenesis, and I think we're halfway through this presentation.